I'll come back with some questions.
Thank you so much, and thank you for the invitation and for your interest. NaDeno is a nanoparticle company, and we have a nanoparticle platform. Our lead candidate is about rethinking drug delivery. So NaDeno is a spin-off from SINTEF in Norway. It's quite similar to RISE, as you have here in Sweden. And they are specialized in developing nanoparticles. Together with the spin-off, it also came a platform that is well protected by IP. We have been part of the BioVenture Hub here in AstraZeneca since spring this year, and we have a lot of collaboration with different institutions and contributors, so that we are now almost ready to go into clinic, planning to go into clinic in a year's time. We have a small team as of today, but experienced people covering the most important aspects of clinical development of the first candidate, and also extensive preclinical knowledge. For example, Ida Mørkær is one of the scientists that worked at SINTEF with this nanoparticle platform. So what are we planning to do? So our lead indication will be within ovarian cancer. Ovarian cancer is a cancer form where tumor cells are shedded off from the tubes or the ovaries into the peritoneal cavity. There they stick to the membrane and start to grow to larger tumors. And then some of these tumors escape from the peritoneal cavity through the lymph system, for example. Today, there is very good treatment for extensive disease. So patients are treated with surgery, followed by chemotherapy, and then maintenance therapy based on their genetic makeup in the different subgroups of patients. But even if you have a complete response after maintenance period, you see relapse of the disease in a large proportion of the patients, more than half of the patients. And there's a hypothesis not invented by us, but by the academic community, that there are remaining micrometastases in the peritoneal cavity that start to grow as soon as the maintenance treatment is gone, and then they can start to develop further. Just to point out this, the reason why it comes back is, as you can see on the figure to the right there, in a larger trial there will be blood vessels. So drugs given IV are able to penetrate the tumor and do the work there. For the smaller tumors, they are not yet vascularized, so systemic drug treatment will not reach the tumor, so they are in a way saved from the systemic treatment with chemotherapy and maintenance. So we are trying to address this problem with the platform, the nanoparticle platform. It's actually a platform. It's a lot of different materials. You can build different nanoparticles, and we have come quite far with the CMC. We are ready for advanced manufacturing of the nanoparticles these days. The nanoparticles we have designed as a lead candidate have encapsulated cabazitaxel, which is a taxane that is one of the two chemotherapies that are involved in the standard of care of this kind of ovarian cancer. There are also some special features with these nanoparticles, and that is very important for the drug to do its work within the peritoneal cavity. I will show you that on the next slide. Also want to say that these nanoparticles, they have a specificity not based on biological features. So they have chemistry that allows them to adhere to the surfaces inside the peritoneal cavity. That is important and one of the main features with this nanoparticles. So you inject it intraperitoneally. That is the only way to reach a high enough concentration of drug to target tumor cells that doesn't have blood vessels. In the cavity, these particles, they are sticky, so they will adhere to the membrane. and they release the drug somewhat over time. This means that the particles in a way reaches the membrane before it starts, before all of the drug has left the particle. This leads to a more even spread of the drug in the peritoneal cavity, and also prolonged exposure of the drug towards the tumor cells. Further, we have seen, based on the tissue makeup in the tumors and the normal tissue in the peritoneal cavity, that there is an increased binding of the nanoparticles where the tumor cells are. We are almost through with the preclinical studies. This is our proof of concept study in a mice model. So to the left you can see intraperitoneally administered cabazitaxel as a free drug, to the left and right. And the small blue dot there, also to the left, is the cabazitaxel in the same concentration given as nanoparticles. So you see a tremendous reduction in the tumor volume in the PACAB or nanoparticle administration. To the right, you see mice treated with either untreated with free IP or with 1 or 2 doses of PACAB 002, which is the name of our drug. With 2 doses of PACAB, all animals survived throughout the course of the study. So you can try to illustrate the landscape here in clinical development in this way. So for macroscopic ovarian cancer, you have quite established standard of care. Surgery, chemotherapy, and maintenance, for example, olaparib from AstraZeneca. And when you give this treatment, you see actually quite good response rates. Up to 70% of patients respond with a PR, partial response, or complete response. But you also see among those patients that have a complete response, around 50% of those will relapse within a year, within 3 years. And as we discussed up front, this is mainly because of this non-addressed micrometastases that are left in the peritoneal cavity without vessels, so they are not treated with systemic therapy. This is just to illustrate how the landscape is right now. So to the left, standard care as of today. In the middle is the development of IV drugs addressing macroscopic ovarian cancer. And to the right is where we are, clinical trials that try to address the micrometastases within the peritoneal cavity. We are planning to start phase 1 or first-in-man trial next summer or in a year's time. That will be a standard 3+3 design of such a trial, and we will be in FIGO3 patients for those that are specifically interested in this. That is the patients that have intraperitoneal tumor and not something outside in distant organs. We will treat with the monotherapy to understand the safety and tolerability of this drug with 2 doses. Of course, the objectives will be safety, PK, recommended dose and tolerability, and also maybe efficacy signals. But so this is a nanoparticle where we have exploited some of the features of this platform. It has this slowed release of drug, allowing exposure over longer time. It has this stickiness that allowed particles to bind to the membrane, so the drug is released at the right place. But we also have platform where we have tested this on several different other drugs. So it's not meaning that you should read this, but up to the left is other drugs where we have made nanoparticles. To the left at the bottom, this picture shows that even if you encapsulate the drug, the mode of action of the drug will not change, and it will not be chemically changed by being in the nanoparticle, so the same function. Also, due to this stickiness, we are able to concentrate the drug in different areas, as you can see up to the right. And you can also dress the surface of the nanoparticles in different ways, so it have different features in different circumstances. There are also some interesting immunological properties, which I cannot discuss here today. Timeline and upcoming milestones. So we are ready for GMP manufacturing. We have been through different steps in that process. We have discussed with authorities in US and Europe, okayed with them, and we are now working with the final part of the GLP tox study and CTA submission in first half of 2026, and a start of clinical trial early second half next year. So this is a very exciting period, we think. So this is an area where a lot of patients doesn't have a lot of good options today. There are no standard care for this micrometastasis, and we do hope that we can make a difference here. So please reach out if you want to discuss. The CEO is over there, and myself here. So thank you so much.
Thank you very much, and please raise your hand if you have any questions. Just curious, one question, maybe not as smart, but why do you think it works on micrometastases but not on metastases?
Well,
how does it? What's the change, so to say?
Well, so if you use chemotherapy intravenously, you will not have high enough doses to treat micrometastasis in the peritoneal cavity. And if you give the drug in the cavity, you cannot expect high enough doses in the general circulation to really kill off larger bulky tumors. Just one more comment to that. So there has been several trials 10 and 20 years back where they actually gave IV treatment with the standard care as of today, and on top of that gave intraperitoneal treatment with the same drugs. They did see good results, PFS and overall survival results, but the toxicity was not acceptable. So therefore it never became a standard care. So our ambition with this drug is we know that the drug works because it's approved and it's on the market, and we know that it works. So it's more, do the nanoparticles hold back the drug in the peritoneal cavity to such an extent that the micrometastases can be killed and still the safety profile be a good one.
Okay.
What is the metabolism or excretion of a drug when you give it
Yeah.
I think you need to.
Yeah. So metabolism and excretion of drugs, yes, when you do that. So when a cabazitaxel is released from the nanoparticles, to put it that way, it will either go into the tissue in the peritoneal membrane, or it will diffuse into the general circulation. So you will find cabazitaxel there, and it will be metabolized as other taxanes. For the nanoparticles, they will either remain in the peritoneal cavity and be degraded over time, or it will be captured by the immune system and transported out of the peritoneal cavity for the metabolism.
Okay.
Okay, thank you very much. How many patients will you enroll into this phase 1?
It will be just a standard 3+3, so I guess around 20 patients. So we hope to move as soon as possible into a randomized setting to really get the efficacy understanding as well for this drug.
And it will be in Europe or?
Yeah, most likely it will be in Europe. We are already in close collaboration or discussions with several hospitals interested in this kind of procedure. So yes.
How about funding? How will you fund this research?
Well, right now we are owned 30% by Sintef Venture and also some family offices. We need to raise money prior to the phase 1 trial. So we are looking into the different options there. But we are very interested in discussing with people that are interested in such a project.
Yeah, I understand. Time flies. We don't have any more time, I'm afraid. So thank you very much for coming.
Thank you.